So good morning, everyone. I'd like to say that it's really a pleasure to be here and meet some old friends and some people that were just getting to know each other. I think it's a hard time for everyone who's trying to push this kind of stuff into the clinics. So that would be very important if we can just share and some experience and speed the process. So I would just present you today some of the things we've been doing in the National Cancer Institute in Rio for the last almost 8 years now. And it's pretty much everything done in Brazil, actually. So we've been doing this for quite a while. And I think it's not necessary, no longer necessary to make an introduction on CARs. Dr. Levine and all the other presenters just presented everything we should know about CARs before starting discussing what I'm about to discuss with you guys. So my only introductory slide would be this one, so that we can see different generations of CAR constructs, right? So all the story started with first generation that would provide only the signal one, or would be the zeta chain. So now we see approved for clinical use in the FDA and commercialization the second generation CARs, either with CD28 or 4-1BB co-stimulation. And you can see also tons of clinical trials using the third generation CARs. So I'd just like you to keep in mind those different configurations of CARs we've been using ever since these second generation CARs. Pretty much the same people in the UPenn have been using for a long time, and that's the one approved by Novartis. And we got it from Dario Campana, who was the original designer for this construct, in two versions, one recognizing CD19 and one recognizing the CD20 antigens, right? And I'll explain why it's important. And we got very concerned on the first reports of on-target of tumor antigen recognition by CARs. This is the result of a third-generation CAR from the NCI that just got one patient killed by recognizing the proper antigen on the surface of lung cells, and was not designed to perform this effect. So we started early considering ways of getting rid of this kind of on-target off-tumor recognition. So we started just, Dr. Levine already presented and mentioned the strategies of using two different CAR molecules in order to make those logic gates. So we started using this configuration of two partially activating CARs, one carrying the signal 1 and the second CAR carrying the signal 2. And that would be a situation where only when you meet the two target molecules you will activate the T cells and have the result. So this has been published already by different groups as proof of concept and very nice papers, basically Michel Sadeline's group at the Memorial. And later we started using this kind of combination that would be an activating CAR and an inhibitory CAR. The activating CAR would be this one we had just discussed, the second generation CAR against CD19. And the inhibitory CAR we just chose a signaling domain that would be derived from one of those inhibitory receptors you can find in every single recently activated T cell. So we'll get there a little bit deeper. And we've been doing several different things as well, just modulating CAR activity with microRNAs. We just started designing some synthetic signaling circuits, and Dr. Levine already mentioned that. And I'll just talk a little bit about these other two strategies we just started developing that would be CAR and gene editing and CARs as a transient way of redirecting lymphocytes toward the tumor, right? So the combination of targets would be always here would be CD19 and CD20, as I already mentioned, and the reasons we already discussed it today. So the CD19 would be a very early precursor B cells, and CD20 would be a marker expressing more differentiated cells. And the idea would be to use as inhibitory signaling domains the stimulating or the signaling domains from CTL4, PD1, LAG3, BTLA, and we have tried several others as well. So this will be pretty much the design of the cars. So this will be in an inhibitory car with the SCFV against CD20. This will be an activating car, and then we have to put a MIG tags in order to detect and distinguish these two molecules, right? And in order to test this system, we just developed a Jurkat cell line that's a regular CD4 cell line. And we just put a reporter system on this cell where there's an NFAT responsive element. So every time this cell gets activated by the TCR, it just glows in the presence of luciferase, since the luciferase is under the control of this responsive element. And on the other side, we have the K560 two cells that have been modified to express each one of the single targets of combination of the targets. So now we can just mix everything and see what happens when you stimulate the cells with the different combinations of antigens, right? So those are the Jurkat cells. So we'll have the version with only the activating CAR and the double positive for activating and inhibitory CARs, and several different inhibitory CARs with the signaling domains from LAG3, PD1, kind of a second generation inhibitory CAR, and the truncated and mutated versions of the PD1 signaling tail. So that would be a way of just ascribing to the PD1 the signaling properties of inhibiting these T cells. So whenever we just put the Jurkat cell with the activating CAR, and you put the target cell either with only the CD19 or double positive, you will always have a very nice activation. This is luciferase activity. And by combining those cells with the different combination of CARs, activating and inhibitory, you see that you can only find a trend to inhibitory function when you use a K562 cells, double positive for CD19 and CD20 when the inhibitory CAR is the PD1. We've been looking to other markers as well, just like CD69, that's a recent activating marker, and then you can see that no difference at all can be observed, right? So we just decided to move it into primary human T cells. And to do so, we decided a long time ago so those cells had been modified with lentiviruses and retroviruses. And a long time ago, we decided to use transposons because that would be much more straightforward. And considering the Brazilian environment, that would be much more straightforward to keep into the clinics as well, right? So this is a system where you have a plasmid with your transgene that's have those ITR domains or repeats in both sides, and then a transposase will recognize it, cut it, and put it in every TA sequence they find into the cell genome. So this is the Sleeping Beauty system. And by doing so, I have some, okay, no problem. So we have described this a few years ago just to express GFP on those cells, and they keep the expression very nicely, so you can expand them a lot for a long time. And this is also true if you want to express CARs, for instance. So you can put the CAR into the transposon, put the transposase along, electroporate the cells, and now you have the expression of this CAR. And to expand those cells, we have decided to use an EBV transformer cell line. That's a B cell transformer, and that cell will be CD19 and CD20 double positive, right? So again, this is a very straightforward doing it. You just push the button and put the cells in culture. We've been doing this for dozens of patients already. So you can see changes in the phenotypes of the cell during the expansion and activation process. And you can see the phenotypes change, and it depends also on the CAR you use, first generation CAR, second generation CAR against CD20, CAR against CD19. So that's something that we've been doing for quite a while, right? And those cells expand beautifully. You can push them just like Dr. Levine showed for many, many weeks. And we are all aware that we should work in this phase here, right? But you can push it forward. And just by re-stimulating them with the EBV transformer cell line. And by doing so, you generate those T cells that won't target any cell that has no target, such as K562 cells. They will target the EBV transformer cell line, so they would be antiviral cell lines that could be used just like mentioned, and they would target cells with the CD19 target in the membrane, such as the NALM6 cell line. So this works very nicely. This is true for cells that express the anti-CD19 CAR, but won't be true for cells that express no CAR at all, right? So by using this system, you can now co-express more than one CAR, right? So you can put 2 CARs in 2 transposomes, and then you have this kind of scenario here. Double negative cells, double positive, single positive for each one of the CARs. And then you can activate these cells with the double positive cells, such as the L3A8 I just mentioned. And then you can just follow up these cells week by week, right? So you can have cells that have only the activating CAR, combination of the 2 CARs activating an inhibitory CAR. And here we'll discuss only about the 20 PD-1 inhibitory CAR. And now you can quantify this 3 population, double positive, single positive for the inhibitory CAR, and single positive for the activating CAR, right? So this is exactly what's shown here. Double positive in green, single positive for the inhibitory CAR in red, and single positive for the activating CAR in blue. If you put only the activating CAR, you see that those cells expand nicely and will get enriched in the CAR positive population. But when you put the combination of the 2 CARs, even though the activating cell has the 2 targets, surprisingly we see the double positive cells expand more. And that's something we see over and over. We have done more than 10 patients, it's always the same. 10 donors, and it's always the same. If you put only the inhibitory CAR, cells will lose the expression of this CAR in a large amount. And that's kind of expected. This CAR shouldn't give any advantage to these cells, right? But any of the first or second generation CAR by itself will enrich. But when you put them together, you'll see the expansion of the single negative or double positive cells, either when they come along with a second generation CAR or a first generation CAR. Oops. Again, a different donor, pretty much same result here. Enrichment and double positive cells. So it seems that putting inhibitory CAR somehow favors the cell that's expressing this CAR in this enrichment or expansion setting. So we went on to use the mutated CAR that had been mutated in the 2 signaling domains of PD-1 of the truncated CAR, inhibitory CAR, that has no signaling tail to see if it holds, right? If the results will be the same. And surprisingly, again, doesn't matter which CAR you put as inhibitory CAR. If it's truncated, mutated, or so, you have always enrichment of the double positive cells. So it kind of, enough to have the binding domain of the CAR against a second antigen to enrich those cells, and doesn't matter if it signals toward the cell or not. It's kind of surprising result as well, right? So maybe it's a way of dampening the activation of the cell and favoring the window of activation, or otherwise a way of stabilizing the synapse. We really don't know what's going on. So we just went on to see what's going on in terms of activity and cytotoxicity using a NALM6 cells that has been transduced to express the CD20 marker. So it will have the 2 markers in the surface. And now you see that whenever you have no CAR or the inhibitory CARs, the lysis curve is very poor, although some killing is observed. If you have only the activating CAR, the lysis curve is much better than whenever you have the inhibitory CAR. So it seems that this inhibitory CAR favors the expansion but not the lysis of the target, right? So we're just trying to figure out what's going on here. Let me move forward to another 2 approaches we're still developing. So the first one would be CAR and gene editing. So we've been using CRISPR. It's something that pretty much everyone knows what it is nowadays. So it's a way of cutting the DNA in specific regions. And now we have been using to try to shut down the CTLA4, PD-1, or LAG3 receptors in primary T cells, right? So we just built the CRISPR, validated it in 293 T cells. That's an easily transfectable cells. And then the plasmid carrying the CRISPR has also GFP, so we can just see the expression of GFP on those cells after electroporation. And we can just check the genome sequencing of the CTLA4 gene and see that's largely edited on those 293 T cells. The same for LAG3 when you use the CRISPR for LAG3, and the same for PD-1 when you use the CRISPR for PD-1 sequences. Of course, mock transfected cells are not edited as expected. And then we decided to move the PD-1 CRISPR into primary PBMCs, so you can see that around 20% of the cells will be edited for the PD-1 sequence, a genomic sequence. And that's not much, but that's exactly where we went. So the setting here to test this would be combining different CRISPRs in the PBMCs that will receive also the CAR by transposase system electroporation, and push those cells in the culture in the presence of the activating cell that would be the EBV transformer cell line, right? And see which one of these gene editing would be enriched in the end of the culture compared to the beginning, to see if we can ascribe to any one of those inhibitory receptors the function of restraining T cell expansion, and if by shutting down this receptor you have a better expansion of those cells, or maybe shutting down combinations of these receptors. And to do so, we took advantage of an approach pretty much similar to have been published by Carl June and Bruce Levine and a group in Philadelphia, where they gene edited by zinc finger nuclease, the CCR5 gene, and gave those cells back to patients infected with HIV. So that would be selective pressure to enrich for edited cells. So we've been doing this in collaboration with Amir Kantanuri with a CRISPR to CCR5. And here you can see edited cells with very low level of addition are put in culture in our very same system here and infected with a CCR5 tropic HIV for the last 5 days, so you can see a rise in the number of the proportion of edited cell lines, cells, right? It's the primary T cells from human donors. So we're just analyzing this, and we are using this system because the activating cell has the ligand for CTL4, the ligand for PD-1, and the ligand for LAG3 as well. So that should be a way of evaluating. So we're just deep sequencing these two scenarios right now to see if we're gaining representative editions of these loci in this moment compared to this moment here. So I promise to show you the data in the next opportunity. And to end the description here, we've been working with the idea of self-limiting the CAR potential off-target effects by transiently expressing the molecule in the effector cells. And to do so, we started working with NK cells for several reasons. And NK cells can kill the target cells by missing self mechanisms that are well known here. But they can also kill cells by Fc receptor loading of antibodies. And Campana's group have shown many years ago that if you express a CAR on NK cells by lentivirus transduction, those cells will be redirected toward the new antigen and kill beautifully the target cells. So when we electroporated the transposon on NK cells, we could never see the CAR being expressed on those cells. So they seem to be intrinsically resistant to transposon gene modification. And that's something that other groups have observed as well. So we decided to take a different approach and use this phenomena, that's phagocytosis, that's the transfer from one molecule from one cell to the other once they interact. And some groups have shown that NK cells could take HLA-G or CCR7 molecules from a donor cell line. So we decided to just start expanding those NK cells in vitro from healthy donors by using those artificial APCs that will expand beautifully the NK cells. So we just end up with a very nice enrichment of NK cells after 4 to 3 weeks of expansion. And those NK cells can be now loaded with the CARs if we use a donor cell that has been built, engineered to express high levels of CARs molecules in the membrane. So now we're using here 20 BB zeta second generation CAR, right? So after just incubating those cells for 2 hours, we can see that most of the NK cells will be loaded with the CAR molecule. And this can be seen here by flow cytometry. So this is the 20 BB zeta CAR being expressed on the surface of NK cells. You can see the red dots here on NK cells that have been loaded after co-cultivation with the donor cell line. So those cells have these red spots representing the CAR molecules in the membrane. And it doesn't matter if you expand cells and get some NKT cells or pure NK cells. Whenever you just go to phagocytosis, around 50% of the cells will be loaded with CAR. And this CAR will be functional, killing the non-sick cell line that has been engineered to express the CD20, and killing also primary blasts of the acute leukemia patients which express the CD20 marker. So we're not just moving this into preclinical models. We have just a couple of preliminary data on SCID mice, and we're just moving this to NSGs, and that should be much more favorable scenario to see this. But you can see that the NK cells will prolong the lifespan of the animals that have been grafted with this leukemia cell that's CD19 positive, but CD20 negative. And it doesn't matter if you have a CAR against CD20 or CD19, so that seems to be a non-specific effect of having NK cells infused. But the clinical score of the animal would be much more delayed if you put the CAR that has the specificity to the leukemia than if you put a non-specific CAR on the NK cells. So we're just moving this into a more refined model to see if this holds in terms of prolonging the lifespan of those animals. So we have this takeoff message. That's a pretty simple way of CAR expanding NK cells, of loading those cells with CAR. That CAR will be, it lasts on the NK cells for 48 to 72 hours at least. And those cells will be specific for the killing of the target cells or leukemia target cells expressing the target, specific target for the CAR molecule, right? So it's a pretty straightforward way of loading effector cells with the CAR molecule without gene modifying them. So I'd just like to acknowledge that people who have supported us with by sharing some reagents and protocols, the people on the lab who performed the work, and the funding agencies. So thank you very much. I'll be happy to take any questions.